Hello, my name is Eugene, and I work as part of Dr. Fink's lab. And for the summer, I'm investigating asynchrony in seizures. So traditionally, the consensus is that seizures are the result of very synchronous neuronal activity. And this makes intuitive sense because for seizure activity, we often observe like a strong rhythm in the brain signal. So it is only natural to think that it is the sum of a collection of neurons doing the exact same thing, producing a signal. And that is true for some case, but recently there are studies that, experimental studies, that show that coherent brain rhythms could actually can emerge from asynchronous neuronal activity. So, for example, here, this is the common view where neurons are firing synchronously, and as you can see, as these dots here represent the firings of neurons, and here neurons are spiking at approximately the same time, producing a regular frequency in the brain signal. And on the other hand, paradoxically, you can see that the neurons are firing rather asynchronously. As a whole, it looks like a little bit of a mess, but still we see a regular brain rhythm emerging. So throughout the summer, we wanted to answer this question, like how do these strong brain rhythms emerge from completely asynchronous neuronal activity? So one way to do this, to study this, is so basically we did a computational model where we could study more in detail what this is, what the mechanism is, so the underpinning mechanism behind it, and what we did was, so to model this, one easy way is to just model each neuron to fire at the same frequency, and basically just changing the phases, and that would constitute like an asynchronous neural activity. But we wanted to push the model a little further and see if when we add more complexity, what would happen. So the complexity that we added, the heterogeneity that we added, was the variability in the intrinsic interspike interval and jitter. So let's explore the first one a little bit. So the intrinsic interspike interval is is basically what it means. It's the interval between the spikes between neurons. So basically, what it is, it determines the frequency of the firing rate of the neuron. So now we can control the variability in the intrinsic interspike interval so we can spread the intrinsic interspike interval with a Gaussian distribution. So the population is the neurons in the population has a intrinsic spike interval that is spread based on the normal distribution that is governed by the parameter sigma ISI, and that would give some complexity to the model. And the other heterogeneity that we introduce is jitter. So once we get the intrinsic ISI for each neuron here, we could also vary the exact spike timing between from the regular expected spike times. So once we do that, we can then go on and convolve it with a biological signal, either a action potential or a postsynaptic potential. Add them all up, and we'll get a brain signal as you can see here. So now, once the data is generated, we can also once we get this signal, brain signals, this data, we could we want to see if which one gives us a coherent brain rhythm, which one could give us a which one gives us brain rhythms and which one doesn't. So for a smaller heterogeneity, so for example here with 5% for both of the heterogeneity, we observe a rather regular brain rhythm emerge, and we get a narrow band oscillation. So basically, we get a very strong frequency component in the brain signal, as opposed to when we have a larger heterogeneity. So for example, here, where both of the parameters are about 50%, we get a more noisier signal. So these two are rather extreme opposites. So we wanted to see what's in between, and this color plot basically does that. So here, the redder the color plot is, the more prominent the frequency, the more it pops, the more it's not just plain noise. So when we do that, we observe that the combined heterogeneity of about 20% gives us a more prominent frequency in the brain signal, as opposed to larger values. In conjunction with that, we also work in collaboration with the University of Michigan to derive an analytical derivation of the power spectrum. And what we found was that, so these plots here are for the special case where the initial where each neuron are initialized so that they are synchronized, but then they gradually desynchronize. So the red curve here is our analytical derivation for this special case, and the blue curve is the numerical simulation that is simulated with this model. And as you can see here, the plots they fit pretty well. So we think we are close to getting an answer. Of course, this is just a special case, so we wanted to have a more general model where everything is asynchronous. So we're still working on that, but we're encouraged by our preliminary results here. So one way to test our model is to compare it against real life phenomenon. So one of them is we find interesting is low voltage fast activity. So these are brain rhythms that are about 20 to 30 hertz that are often observed at the start of seizures. And what's interesting about low voltage fast activity is that they feature narrow band oscillations. So we can see this very strong frequency component here, but they're thought to be the result of completely asynchronous neuronal activity. So this is an interesting case. Before this, it's a complete mystery. We don't really know what happens, what's going on here, but we think our model could explain this. So to test this, we created a biophysical model. So we actually just borrowed the model that we used in a previous study and basically simulated the key neuron here that is thought to be responsible for low voltage fast activity. And we basically decoupled them and stimulated them so that they would generate firings that are asynchronous and therefore induce an asynchronous postsynaptic potentials in the larger cells. So here our results showed that yes, we get asynchronous neuronal spikings, but we also get this really nice narrow band oscillation at just about 25 hertz, consistent with the low voltage fast activity that we wanted to test against. So this is really encouraging result. So in a nutshell, this work suggests a new mechanism for the generation of brain rhythms, and these brain rhythms can emerge completely from asynchronous neuronal activity. And we predict that this asynchronous networks can generate narrow band oscillations with up to 20% combined heterogeneity, sigma jitter, sigma ISI in the neuronal spike timing. Also, as a nice proof of concept, we use a realistic biophysical simulation to demonstrate that an important brain rhythm like low voltage fast activity could emerge from asynchronous neuronal spiking. So thank you.